The following is an overview of my EDUC 522 unit plan on genetics. This unit was planned for high school biology, grade level 9 to 10, for a duration of 8 weeks, including 4 days of instruction and a block period using the textbook Prentice Hall Biology 2007. This unit combines levels of Bloom's taxonomy and Gardner's multiple intelligences to transform dull and boring traditional pedagogy into engaging lessons with integration of technology. This unit is designed to meet the next generation science standards with connections to Common Core standards, as well as ISTE standards for technology. Objectives for this unit are as follows. Students will be able to know the structure and function of DNA and RNA molecules, explain central dogma, explain how genetic information is passed from generation to generation, apply rules of probability to principles of genetic inheritance using Punnett squares to complete their crosses, draw and describe the phases of meiosis and compare and contrast this process with mitosis, explain how genetics can be used to better understand human inheritance, and discuss and defend a position on ethical issues concerning genetic testing and bioengineering. Students will engage in several projects and activities throughout this unit. During week 1, students will learn about DNA structure and function by creating DNA origami models, creating a DNA rhyme using a beat on the Bongos app, and extract their own DNA following an instructional video from Nova. Students will also explore the structure of RNA and its role in protein synthesis as explained in Central Dogma. Students will create a flowchart using the MindMaple app, write a poem in iambic pentameter using the Pages app, and simulate the various steps involved in protein synthesis using a model kit. In weeks 3 and 4, students will compare and contrast the processes of mitosis and meiosis through various activities engaging different learning styles. Students will complete an onion cell lab with microscopy and chromosome kits, choreograph and make music for a mitosis-meiosis dance using iMovie and GarageBand, and create several graphic organizers using mobile apps from Read, Write, Think.
Okay, so in prophase, what happens to your membrane?
It
Disappears. Good.
Then you get the tetrad.
Okay, forms that tetrad. Crossing over occurs. Okay, you guys already crossed over. Good.
And then
Now we're going to do metaphase.
During weeks 5 and 6, students will learn how to predict and solve genetic crosses using a step-by-step model. Students will then form collaborative multiple intelligence groups to create a presentation to teach their problem-solving method to the class. Presentations include a screencast made using Explain Everything, statistical analysis and charts using Numbers app, fruit fly genetics report using Keynote, and a telenovela explaining the use of blood typing in an infidelity case using iMovie and GarageBand. GeneScreen is another app used to explore genetic crosses and Punnett squares that will be used in this unit for students to practice Punnett square crosses and also learn about different diseases related to certain ethnic groups located around the world. In weeks 7 to 8, the unit will conclude with a debate on different issues in genetic ethics as students learn about the implications of genetic testing and the Human Genome Project. Students will debate their positions on genetic privacy laws and pre-screening of embryos. The debate will be followed with a reflective discussion and essay assessing student growth during the debate process. Integration of technology was used to provide QR codes for students to scan for various research websites. Student learning outcomes from this unit are as follows: remembering key terms and content matter, understanding and explaining main concepts, applying concepts and skills to real-world observations and experiences, analyzing and explaining data, evaluating and discussing the moral implications of advancements in science, and creating lessons for peer-to-peer teaching based on MI's strengths. Throughout the duration of this course and the process of preparing this unit plan, I have learned to create differentiated lesson plans, evaluate student progress using authentic assessments, analyze student strengths and weaknesses, apply knowledge gained from assessments to modify and improve instruction, understand students on a holistic level, and also remembering that all students have the potential to succeed, and understanding their multiple intelligence greatly helped me in that process and challenged me to see students and teaching in a whole new way.